The first thing we're going to talk about today is, sort of go through a little bit of an introduction to how the course is actually going to work, and present some of the ideas and sort of the goals for the next, 6 days together during this workshop. Before we get too into this, I want to make sure that every single person in this workshop is already on the Slack. Scott has done a great job of just making sure everyone should have gotten invited. If you did not or did not see the invitation, please go to kursosomnilab.org/get-help and make sure that you join the #2021 workshop announcements. There, in the bottom of that channel, there's a link to a few other help channels. If you have questions at any time during the lecture, I actually strongly recommend, there'll be some points where we stop and give you an opportunity to unmute yourself. But I think actually the best thing to do is to make sure that you ask questions on Slack and not in the Zoom chat. The Zoom chat gets erased every time at the end of the call, but oftentimes the Slack chat actually is persistent, allows for multiple people to respond, has threads, people can post links much more easily. So please use the help channels there very aggressively. They're broken out into math help, coding help, and data help. And sort of, a lot of questions I imagine are probably going to be math help. And so I would just maybe sort of use that as sort of your central sort of place to ask questions during the workshop. I also, before we get started, would like to acknowledge the wonderful cohort of TAs who have agreed to help out and make sure that this workshop gets run smoothly. We've got a slew of students from across all different levels, undergraduates, graduate students, a few staff scientists from our Genome Center are here with us. We have Smitha. I'm really excited. And I think, 100% this workshop could not work without our TAs, because they're the ones who you're going to end up asking a lot of questions to. They're going to be checking in, on your groups, making sure you answer questions. And so, 100% want to make sure that they get all the recognition that they deserve here. Okay, great. So now I'm going to give you a little bit of an overview of, why we do this course and how we sort of think about the goals and objectives of this course. So everyone is hopefully, is excited about the fact that, in the past 15 years or so, there's been an explosion in access to large biomedical data sets. We're going to focus a lot on single-cell RNA sequencing in this course. But actually, I think a lot of the principles and ideas that we teach apply to a large number of different kinds of data. You can have single-cell RNA sequencing, you can have attack sequencing, large longitudinal patient records, fMRI data. A lot of the ideas that we're teaching, I think, apply to a lot of them and leverage some fundamental properties that are shared across data modalities. When I say modality, I mean, specific thing that you're measuring. But of course, the biggest thing that we're excited about here is the single-cell revolution that sort of happened in around 2015. This is an image from the DropSeq website. You can see here, I think, a really exciting example of, so this is a microfluidic chamber. If you're ever wondering how droplet single cell technology works, like 10x works, there's a chamber that's about, I think this is about 100 microns in width. And on the bottom here, you can see a cell sort of entering, this is one capture event over and over again, entering this chamber. It's in a solution of media and oil, and then encountering a flow of beads and lysis buffers. This large thing here is a bead coated with primers to actually generate a cDNA library from that cell, and then getting pinched off in this chamber into individual droplets. And so you can see sort of droplets go out to the right. This is sort of the input to your single cell library preparation, is how you sort of get data at the end of the day. One of the things that this has sort of led to happen is that, people are starting to do really interesting kinds of biological experiments, picking their favorite kinds of model organisms like we saw earlier, applying a bunch of different kinds of computational techniques to generate really exciting plots and insights into their data, and then hopefully at the end get a high impact paper. And I think, we've sort of seen a trend over the past few years where, especially in biological systems, having some single cell aspect to your investigation ends up becoming an increasingly important part of getting some of these high impact papers. And one of the things that may not be entirely clear, I think, to the audience that we're sort of targeting for this workshop is exactly what's happening in this computation part in the middle. I am one of those people. I have an undergraduate degree in microbiology. I started out my PhD doing pipetting, and I know what it's like to sort of have to have this biological background, but then try to understand, what actually is going on at the computational side. And understanding that that can actually be incredibly complicated, and that sometimes the training in biology is not necessarily the training that you need to understand, all of the machine learning or algebra or probability theory that you might need to have in order to really grasp the sort of computational foundations that lead to the insights that you might find in a publication. It doesn't really help that when you go and read the methods for these computational methods, that it is an entirely different language. It's literally, a lot of it is Greek letters that you maybe don't know the foundation for. And I want to just be totally clear that we are not blaming other groups for this at all. This is actually an excerpt from a manuscript from our lab that was published a couple of years ago, or I guess just about a little over a year ago. Fate, this dimensionality reduction algorithm, we'll talk about in the coming lectures. I know really how intimidating seeing some of this notation can be, especially if you're not familiar with it. And one of the things that we really want to do is sort of address sort of this feeling that you may have, like you look at these papers and you feel sort of like you're just getting a hose of information straight to the face, and that there's like no easy way to understand or sort of grasp onto sort of what's happening. And so we want to sort of give you some of the foundational tools to grasp those methods, to understand what are the actual principles that they're basing, methods are basing their assumptions on. And to start, I want to just clarify, what exactly do we mean when we say machine learning? I think that this is a phrase that gets sort of thrown around a lot. There's a lot of buzzwords for, but actually clarifying, like, what do we mean by machine learning, and maybe how is it different from statistics? And so when we talk about machine learning in this workshop, we're really talking just about a very generic process of learning and identifying patterns in data. And I know that that sounds vague, and that's intentional. There are a lot of things that are machine learning. One of the things I think that you learn as soon as you start to study, machine learning, is that actually there's a lot of statistical methods, things you might think of like ordinary least squares regression, just linear regression, fitting a linear trend line to your data, can be thought of as machine learning. It's really hard to come up with a definition of machine learning that doesn't include some of these really simple, you might have thought of as statistical processes within that umbrella. And oftentimes you'll hear when people talk about machine learning, they'll break it down into two kinds of categories. The first one is supervised learning. So supervised learning is this problem where you have a bunch of labeled data and you want to label some new data. So like a classic example of this might be you have a bunch of images of different kinds of fruit, and maybe you use them as input to your model, and you know that the model says that this is a strawberry. This is a strawberry ahead of time. And so you're sort of enforcing this model over time to show it new examples where you know the label, and then telling it what is exactly the classification that you want, so that you have all of your images of strawberries getting grouped together with other strawberries, and all of your images of bananas getting grouped together with other bananas. And then the goal is that now, let's say you have a new image of a fruit, and you don't exactly know what it is. You don't have a label for it. You want the computer to be able to just sort of look at the model to be able to look at that image and then say, this is a strawberry. So one of the this is, one of the important things here is that for all of these images, we had labels. We knew that they were strawberries or that they were bananas. And so this is, whenever you hear supervised learning, think pre-labeled data. But there's also a category called unsupervised learning. And unsupervised learning is actually one of the specialties of the work that we do in the Krishnaswamy lab. And the goal of unsupervised learning is where you have some data, but you don't necessarily have labels for it. You just want to be able to identify some underlying principles in the data and then use that to organize it for you, and to organize this in a quantifiable numerical way. A sort of classic example of unsupervised learning might be principal components analysis. And so you might, have some image, you give it to your model, and it gives you out a vector representation of it. These numbers maybe don't mean anything implicitly. That doesn't say that these are cherries. But the idea is that if you are basing your model off of some of the underlying principles in the data, if you, feed this model, multiple sets of data points, you find that data observations that have similar kinds of properties, have similar vector or like numerical representations on the output of your model, and are distinct from ones that have some differences. So this is sort of a very, intuitive explanation of the difference between them. But I also want to make it clear that we can sort of quantify and use notation to communicate the exact same idea. So in supervised learning, I said you have labeled data and you want to label new data. This is the same thing as learning a function f of x. So f being a function, x being your data, and the output of that function is some variable y. And the key in unsupervised learning is that you know all of the values of y for some of the samples of x. So for example, we knew the values strawberry for some set of images of strawberries, and we knew the value banana for some values of bananas, for some images of bananas rather. And in unsupervised learning, what we have is a bunch of unlabeled data that we want to organize. And so we call this, the numerical representation and embedding, where we want to learn an embedding of x to y, where x is some, set of values, and we're going to clarify exactly what this Rn means in a couple of slides, and y is some other collection of values where n is much, much greater than m. So x has a dimensionality much greater than y. And so, for example, x for these images, you might have 1,000 by 1,000 pixels. So you have actually 1 million values for x, but you actually want dimensions. For example, I was talking about PCA, principal components analysis. You only want to know PC1 and PC2. So there you have 1 million is much, much greater than 2. And so this is a lower dimensional representation of the data that might be easier to interpret. And if this doesn't make sense right now, that's fine. There's a lot of concepts that maybe we're going to explain over and over again. And please feel free to ask questions on Slack, and then we'll try to reinforce over the coming days. And I'm realizing I unfortunately gave the answer to this question earlier, but we're again just going to make sure that poll everywhere is working for everybody. So I just want everyone here to go and answer. I'm actually going to hide the. I want to hide the answers here until. I'm going to give it a little bit. So I'm going to wait. I think probably just about 20 seconds, and then see sort of what people think, and then go over the answer to the question. So let's see. Someone had asked if n and m were the wrong. So I guess the idea is, I don't remember exactly what it said on the last slide, but what it should say is that x is in Rn and that n is bigger than m. So you should have more dimensions in your data than you have in the embedding space. And I think that is what the slide said. So I think, yeah, maybe we can clarify that on Slack. Okay, great. So we're going to share responses. Interesting. Okay. So 57% of you said that it was an example of supervised machine learning, and 42% of you said it was an example of unsupervised learning. So linear regression is an example of supervised learning, because when you do linear regression, you want to learn a function of x to y, right? So you have two lines, and you want to map x to y. And in order to do that, you have to know some points, x and y, and then you fit a line that minimizes the residual between that line and the data. But so you have to know some values of y for some values of x, so that when you add new points, that you can what the values were. You have to be able to actually fit the line to your data, right? If you have a scatter plot, you know the values of y for some values of x. So linear regression is an example of supervised machine learning. So what about clustering? So is clustering an example of supervised or unsupervised learning? I'm not sure why. I thought these all were closed by default, but I see we've got about 70 responses. And wait till we hit. Yeah, this I think should be enough to see. Great. Okay. Clustering, everyone got right. I'm realizing actually that one was on one of the earlier slides, so I'm glad. But yes, clustering is an example of unsupervised learning. And that's because when you have a data set, let's say you do your single cell sequencing, the sequencer doesn't tell you what cluster a cell belongs to. You have to learn that from the data. So clustering is an example of unsupervised machine learning. And if you have any questions about this, please, the math help channel on Slack is a perfect case. If you didn't understand that explanation of why linear regression is supervised, 100% our TAs are very able and willing to help you in the chat. So with that, this is sort of like the first idea, how we want to teach things in this course. We want to give you sort of some intuitive understandings of things. We want to then present to you maybe some more formal definitions of what they're doing, make sure that you understand things. And there's also going to be coding notebooks after the lecture that should help you sort of cement and solidify the principles that we're teaching during these lectures. I want to make sure everyone is aware. I mean, I think most people are aware, but just make sure if you go to krishnaswamilab.org/workshop, you can find a course schedule here. These dates, of course, are outdated and from our May workshop, but you'll find an updated schedule there. I've also made it clear on that website, it will say 2020 version if the slides have not been updated since 2020, and some things will get moved around. I also just want to clarify one point. We sort of made a change from what was communicated in our last set of emails. The workshop is the office hours are going to end at 5 p.m. Eastern time. And so there's going to be, after the lecture, a 90-minute office hour session, a 30-minute break, and then another 90-minute office hour session. And then it's going to be done for the day. Always, you can post messages on Slack. I know people here are from very different time zones all around the world. You do not have to come to the office hours. The lectures are being recorded. We really want to make sure that this accommodates people's schedules, especially during COVID when we know things are a little bit unpredictable. Slack will always be around. I think you'll find Scott and I will answer things almost any time when we're awake, and then, or at least during reasonable hours while we're awake, and hope that you can ask questions there. Great. Go next. Okay. I also just want to address one point. So we're doing all of the workshop in Python, all of the coding exercises. This is not because we think that Python is fundamentally better than R. It's because we're trying to teach you machine learning fundamentals. And Python is actually the foundational language for a large number of machine learning tools. So there are really great packages for numerical computing and data organization that are implemented in Python that work very well for large data sets in a way that doesn't really work as well in R. There's also really great, there's a whole machine learning toolkit called scikit-learn that has a bunch of models like your linear regression, logistic regression, all sorts of clustering methods implemented, and we're going to use heavily throughout the workshop. We also are going to spend a couple of days talking about neural networks, and of course the first class neural network libraries, TensorFlow and PyTorch, are all in Python. And there are actually also really great tools for analyzing single cell data in Python. So ScanPy from the Tice lab and scprep is a software package from our lab. Really sort of give you a lot of the sort of similar tools that you might want from, for example, Seurat or different tools from Bioconductor available in Python. So we actually do all of our single cell analysis in the lab in Python and really encourage people to sort of get their toes wet with that language. We're also going to use Google Colaboratory a lot. This is essentially a notebook where you can run Python code for free. It's from Google. You just need a Google account to access it. There's also a pro subscription available for people. If you really like using Colab, you can pay, I think it's like $10 a month and get access to sort of upgraded server resources. And that way you don't have to run everything on your laptop, which I think can be a little bit hard for people. And the nice thing is that Colab is sort of guaranteed to be a fresh instance for every single person. We don't want to spend half a day installing, all of these different tools on your laptops. We'd rather get you started immediately, knowing that the code is going to run properly. I also just want to address that during the office hours, we really want to emphasize sort of team-based learning. So we've offered this workshop a few times now, and one of the most important aspects for learning how to program and sort of understand the concepts in the course is going to be talking to and working with each other. After the lecture, we're going to break out into Zoom rooms. Those are going to be sort of your group work rooms. We really highly encourage you to have one person share a screen, show what they're doing, talk about it, ask each other questions. I'm sure things are not going to work the first time, and you're going to have some questions like, should our cutoff be X or should it be Y? And I think your teammates are a really great resource for that. The last thing that we want is people to be sort of disconnected and in their own room, or not really knowing, not feeling like they have a group to talk to. If that ever happens and you're worried that like, hey, my group's not here, I'm looking for my group, please come into the room and let me know. We will 100% take care of that very quickly and make sure that you have a team to work with throughout the office hours. And this may be a little bit more fluid as people sort of solidify what time they like to work. We can be very flexible with the teams, but we don't want people sort of in a room on their own. We're also, one of the instructions we gave to the TAs before this, and what we're going to sort of let you know now, is that the way that we're going to teach is, rather than giving you a fish, we're going to teach you how to fish. And so you're going to have a lot, we expect a lot of people are going to have problems where there's a Python error that comes up and they don't understand what it means. And this is 100% normal. This happens to everyone. I sometimes have Python errors. I literally just yesterday messaged Scott about something that I didn't quite understand what it means. And I had to, get some help parsing it. Oh, this is the important section of it. This is the thing that you need to go do. And so the TAs are really going to help you understand what the error actually means so you can start to fix it on your own. Because you can't, we want to make sure people are not just saying, what is the correct answer here, but rather, how do I arrive at the correct answer given the errors that I'm getting? And so with that, one of the things that we're going to teach you how to do is how to Google for things. Like we use Google every single day when we're trying to fix software programming issues. And so we're, don't be surprised if your TA says, Google this part of that error message. That's an important part of helping you understand all the resources that are out there for fixing and debugging code. We're also going to show documentation and encourage you to look for, hey, this function is saying that you used it incorrectly. How do you go and figure out what it wanted you to do? And so, you're going to maybe find some people are going to show you the documentation and highlight the section of the text that you need to know in order to actually grasp, like, this is the problem that you had. Here's how you could next time go and see what the correct way to do it was. I also want to just mention that a lot of people are interested in analyzing their own data in this workshop. That is super awesome. We're very enthusiastic about people using this as an opportunity to dip their toes into their own data sets. There are a few ways that you can get help with that. Scott has done an amazing job of making sure that there are some notebooks available on the workshop website that actually give you a template for loading your own data in Google Colab and running some of the code that we had in the course. If you are concerned about your data being on Google servers, I understand this is a global audience and some people have restrictions on where their data is allowed to go. I understand that. Unfortunately, it's going to be really hard for us to troubleshoot some of those technical problems if it's not on Colab. We still will give it a try. There are also two places to go if you have questions. So one is this Bring Your Own Data Help channel, or it might be just called Data Help now. And the other is that we have Smitha here, who is a professor who has spent her career as a faculty member thinking about single cell data. And she's very generously agreed to make herself available to anybody who has questions about their data. I don't know, maybe Smitha, do you want to just mention a little bit about how they should get in touch with you?
Yeah, you can get in touch with me by Slack or by email. So usually how it's worked in prior workshops is somebody has a data set that they want to analyze, and they have some usually goal in mind. Sometimes they don't have a goal in mind and they just want to consult with me about what machine learning method that they should be using to get the best results, or brainstorm with me about what else and what other kinds of insight they can get from their data. So a lot of it is kind of conceptualizing and brainstorming together, and often people end up having good ideas as they leave their workshop about what kinds of things they want to do with their data, and maybe examples of other papers where they've done that and things like that. So it's pretty open-ended, free form. So and I usually schedule it on my own personal Zoom, sort of like a 20 to 30-minute session where I'm chatting with people.
Yeah, I highly encourage everyone to take advantage of that opportunity. That is going to be sort of Smitha's goal during the office hour sessions, is to have those during the afternoons. I mean, maybe not every day, but just make sure you take advantage of it. Last time I think people waited a little bit too long, and so on the last days of the workshop, there was a bunch of people who wanted advice. If you think you want advice, reach out to Smita sooner rather than later. There, two more details before we get back into the technical content. So one of them is that we have on our so I know that there's this workshop website. There's also a workshop materials website. This link you should have from your email. You can also just type this in here. It's not available on the front page of the website. This is a bunch of information that's really useful and organized by day and topic to help you sort of grasp some things. I think, it's really important that you don't just use these lectures and think like, oh, if I listen to the lectures, I'll understand everything. You're going to have to go and look at other people explain topics and ideas, and we've spent a while actually going through and making sure that every single topic that we talk about in the class has some other sort of either YouTube video or blog posts explaining them. And if you have other suggestions for things that you think should be on here, that's 100% something that you should let us know on Slack. So please feel free to tag me or Scott at Daniel Burkhardt or at Scott Gigante, and we can help you with that. We also are on Twitter, and really, I think a lot of people have found out about this workshop through Twitter. You can tag our lab at Krishnaswamy Lab. We're using the hashtag YaleML, help other people learn about the workshop and sort of make sure that this is something that's available, people get excited about. So 100%, love communication over Twitter as well. So with that, Smita, do you want to request control of the screen, and then I'll hand things off to you?
Okay, requested. Okay, so hopefully everyone can hear me. Just let me know in chat if you're having any problems with that. I'm going to talk a little bit about representing data. So the essence of unsupervised learning, which is extremely useful in single cell analysis, largely because we're measuring these cells because we don't know what's going on and we don't have the answers and we want an unbiased look. And so unsupervised learning is very concerned with learning a representation of data that you can interpret, that you can extract structure from. But data already has a representation, so when it comes off the machine, you have a representation of data as reads, and then you might turn that representation into a representation of data that's a counts matrix. So one of the keys to unsupervised learning is that you're taking a representation that might not be as friendly for extracting structure and turning it into a representation that becomes friendlier for extracting structure. So I'm going to go over kind of the basics of that now. And I'm going to cover one kind of representation that turns out to be enormously important for a lot of the machine learning unsupervised methods that we're talking about, and that's to represent data as a graph, even if you don't think of the data originally as coming from a graph. Okay, there we go. Now it's going too fast. Okay, there we go. So in single cell data, you have each cell encoded as a vector of measurements. And these measurements usually correspond to, for example, in single cell RNA sequencing, they correspond to mRNA counts. And there usually has been some pre-processing to get it to that state. And Dan will talk a little bit more about the pre-processing in the next component of this lecture. But nevertheless, you can think of each cell as a row in this matrix, and it's a vector of measurements, which are all counts. So an observation is a row, and the whole matrix is a set of cells. And so each row is a cell and each column is a gene. So this is sort of the standard representation we think of starting with, which is an observation by feature representation, and it's a matrix. So matrices turn out to be really important for all kinds of machine learning, supervised and unsupervised. And a lot of unsupervised and supervised learning amount to changing matrices to other matrices in certain ways. So whenever somebody asks me if I took one math class, what should I take? Definitely linear algebra. But nevertheless, I think you guys are all familiar with this kind of a matrix, which is an observation by feature matrix. And so if you have many single cell data sets, this can even become what's called a tensor or a collection of matrices. So you have your cell by gene matrix that represents each sample that you have, and then you can have many samples as well. We won't be talking about handling an extensive amount of samples in this class, but we will be talking about how to compare between 2 or 3 different samples of this kind of data. When we talk about our data being a high dimensional matrix or our data having high dimension, what we're talking about is there being a number of columns here. So each column is a gene that's measured. And in another kind of single cell RNA sequencing data, like ATAC-seq data, it could be different. It could be that each column is chromatin location. The observations are cells and single cell RNA sequencing data, but on the first slide that Dan showed you, he was also showing some other kinds of data. For example, fMRI data, where each row was a different unit that was being measured, like a brain voxel. Generally speaking, a lot of the techniques that we talk about in this workshop are going to be useful for a lot of different kinds of high dimensional, high throughput data. So high dimensional means that you have a lot of features you're measuring. High throughput means you have a lot of observations. And these are like fundamental characteristics of single cell data that basically ushered in the use of sophisticated machine learning. It's important to actually remember that a lot of the methods that we cover, they're more sensitive to how many cells there are, not so much how many genes or features there are. And there's a specific reason for that, and you'll see it in a second. When we turn this into a cell-by-cell graph, it'll become more very apparent that the number of cells, in a sense, controls the runtime and complexity of some of the methods that we go over. And so whenever you have high dimensional data, meaning you have more than one dimension, you can actually think of it as a point in the corresponding dimensional space. So let's say you were measuring just 2 genes. So you had probes for 2 genes and you're measuring 2 genes in every cell. So you can think about that cell as a data point that lives in a 2-dimensional Euclidean space, sometimes called R2. So the x-axis of this shows the first feature and the y-axis of this shows the count on the second feature. So here there was 1 unit of your first feature and 2 units of your second feature. And so you can readily think about this as a data point that lies in an n-dimensional space for n dimensions. So this gets you thinking about this kind of observational data as a point cloud in n-dimensional space. I'm trying to advance this. So the order that you actually put these axes in it actually doesn't turn out to be very important, some of the things we're doing, but one of your axes will always control the first column and the other axis will always control the second column. Of course, if you have 3-dimensional data, you'll go into the z-axis, x, y, z. And pretty soon you'll see that it's hard to think about more dimensions than 3 because you can't visualize it. And this leads to the idea of trying to reduce the dimensionality down to 2 dimensions or maybe 3 dimensions so that you can visualize the data. And we'll be talking about that a little bit or a lot tomorrow. Another way to go, so one of the other ways to represent the data is actually based on the relationships between these data points and their spatial representations. So here you see that you have 3 data points, A sub 0, A sub 1, and A sub 2, and these are visualized as 3 different points spatially, and you can think about the relationship between these points. For example, how far is a sub 0 from a sub 1? So a sub 0 is at 1, 2, a sub 1 is at 2, 4, and you can use your favorite type of distance that you learned in 4th or 5th grade, Euclidean distance, to see how far they are. So what does distance actually mean here? It actually means that the measurements are pretty dissimilar if the distance is high. So if the distance is low, it means that the measurements are similar. So the distance is a proxy for dissimilarity between these data points. Of course, here's another example where you're looking at the distance between A0 and A2. And so from these two distance measures, it's clear that A1 and A0 are more similar to each other than A0 and A2 are. And so this gives you an idea of the relationships. There are other kinds of distances that you can measure the dissimilarity between your data points. So one measurement that you might have heard of are Manhattan distances. Manhattan distances assume that you can only move along certain grid lines, so that you have to go down a Manhattan Avenue and turn rather than cutting through buildings. This is a valid kind of distance. So the Manhattan distance between these 2 points, A1 and A0, is 3. So now you see that A1 and A0 under Manhattan distance are the same distance apart as A0 and A2, which is also 3. So the amount of dissimilarity you get is actually dependent on what kind of distance you use. And so this is something that you'll want to think about. How do you measure dissimilarity between points? There are other measures of distance. There's and measures of inverse distance or similarity. For example, there's cosine distance, and there's Mahalanobis or variance corrected distances. And this can determine the entire relationship between your data points. So anything can be a distance if it satisfies these 3 properties. So it has to be symmetric. So the distance from A0 to A1 would have to be the same as the distance from A1 to A0. And which is true for Euclidean distance as well as Manhattan distance. You're just walking, starting at the other point, for example, in Manhattan distance. It has to be non-negative, so you can't have negative distance between 2 points, because what would that mean semantically? It would mean something like it takes negative effort, and it wouldn't make sense. And then the third criteria something has for being a distance is that it has to follow the triangle inequality. This is the only one that might be a little bit non-obvious. The triangle inequality just says that the distance from a point A to a point C is less than or equal to the distance from A to B and B to C. So if you think about it, you could always go from A to C by going from A to B and then going from B to C. So the actual distance can't be any longer than that path. So that's what the triangle inequality is saying. So it's going to be equal to it if the distance from A to C, or here the distance from the distance between these 3 points, which you can think of as A, B, and C, where this segment has length x, this segment has length y, is equivalent to the segment of length z. It's going to be strictly less than it if there is a more acute angle between x and y. And this is what the triangle inequality is saying, because it's saying it has to be equal or less than, and that's sort of a visual view of it. But based on this, you can use any distance that satisfies these 3 properties, which is also called a metric, in order to understand how similar or dissimilar your points are very precisely. So, any questions? I just heard a noise. So what you can do based on distances is go from a representation of your data that is encoded as observations by features to similarities between points. And this kind of representation is also called a graph. In a graph, each vertex or point represents an observation, and edges are these connections between these points. And I don't know exactly why it's called an edge. People sometimes, when they first learn about graphs, that's why they're called edges instead of nodes and connections, but that's usually the terminology. They're even denoted E. The edges are the lines that you see that are connecting pairs of vertices. In a graph, you can have weights, meaning strengths of edges. So things that are very similar could be highly weighted. Things that are not very similar could be lowly weighted. If you don't have weights, the basic idea is things that are connected are similar, and things that are not connected are not directly similar. So this kind of graph that has no weighting is called a binary graph. But distances that we just talked about can help us go from a tabular representation of data to a graph representation of data. And the basic idea is you connect things that have low distance or high similarity. Or you weight things with low distance or high similarity higher. So here's sort of an example of how you go from the data matrix to a graph. So notice that a graph is basically defined as weighted edges that go between points, between pairs of points. So now both the rows and columns have observation indications on them. So point A0, A1, A2 are both on the rows and on the columns. And what inside of the matrix are no longer counts, but rather distances between those points. So any point has distance 0 from itself. So you see zeros along this diagonal. But from A0 to A1, you have square root 5 if you use Euclidean distance. And that's going to be the same as A1 to A0 because of the symmetry of distances. So you see the symmetry along the diagonal in this kind of distance matrix. So you can think of this as the inverse of what's called an adjacency matrix that's usually used to represent a graph. So usually in a graph, things with small distances have high weights or high adjacency scores, and things with low distances or high distances have low similarity. So usually graphs are actually represented by matrices that look more like this, where things that are close, like points to each other, have high weights. So in this graph, there's high weights for self edges and low weights for things that are farther away. So the farthest away are A0 and, for example, here, A2. So these have low weights. So the way you get from distances to similarities is actually pretty well studied. And one of the ways you can get from distances to similarities or affinities, that's another term for similarity, is to take your distance and put it through what's called a kernel function. Notice these are examples of many kernel functions. And notice they all have this property where they reach their maximum value at 0. And anything after that, anything positive, which all distances are, has a lower value than 0. And so they have this kind of inverting quality. So if you put in a high distance into a kernel, you'll get a low similarity. If you put in 0 or perfectly similar no distance, you get a high similarity. So the basic idea for getting from distances to similarities, which directly describe the edge weights in a graph, are to take your distance and put it through one of these functions, with the most common one being the Gaussian kernel. Here you have kind of a rather wide Gaussian kernel. And if you put it through this Gaussian kernel, depending on the bandwidth of the kernel is going to be a parameter that you play with in this kind of graph model. It's going to give you a similarity that's scaled. So here I actually did that. I put the distances that you saw here through a Gaussian kernel, which is describing a normal distribution, with mean 0 and standard deviation 1. And when I do that, these are the affinities that I get. I could and I would get completely different affinities from this. But now what I've done is I've taken this data and I've re-represented it as a graph. The weights on the graph are given by these off-diagonal entries in this adjacency or affinity matrix. And now I have an idea of how the points are all related to each other. Now notice that in this representation, I no longer have the features. The features aren't explicitly in this representation. But it turns out you can use derivations of this representation to understand what's going on with the features. This is the reason why a lot of the unsupervised machine learning algorithms are more sensitive to the number of observations than they are to the number of features, and would work just as well with 10 features as they do with 20,000, as in single cell RNA sequencing. Okay. Modification of this graph. So these are fully connected graphs. They just have different weights on each of the edges. A modification of that is to zero out some of those edges, especially the small ones with low weights. And a really common way to do that is using what's called a nearest neighbors graph. A nearest neighbors graph will only have connections between a vertex and its k nearest neighbors. It could still be weighted. So for here, for example, if we did a one nearest neighbors graph, then we'd zero out the connection between A0 and A2 because A0 is most connected to A1 and vice versa. And so in this row, we'd zero this one out. But A2 still has distances, and it turns out A2 is closer to A1. So you zero out the A2, A0 edge. So that leads to a sparser representation, which is good computationally. But the other thing you'll start noticing, so here is a sort of semi-realistic data set where we sample data that's following some trend. Here it's following the Swiss roll-like trend. And you'll see Swiss rolls are used a lot when people are thinking about the manifold or the shape of the data. And here, if you sample points along the Swiss roll and you connect every point to its 4 or 5 nearest neighbors, you see that you kind of get this mesh here that really follows the shape of the data. And that's why the nearest neighbors graphs are useful, and so are their softer versions, the fully connected weighted graph. They're really, in some sense, emphasizing the main shape or the paths in the data. And so a lot of the algorithms you'll see in this class will be built off of affinity matrices or their sparsified equivalent, which is a nearest neighbors graph. Does everyone understand this point? Any questions on it? There might be questions in the Slack. I'm not like actively looking at the Slack. I'll be sure to answer. Somebody asked, when choosing a distance metric, do you want to maximize or minimize a factor, like the affinity graph that was presented in lecture earlier? So usually one thing that can turn out to be important is to have a lot of variation in your data. So the distance, usually people use one of a few different distances, like Euclidean or cosine distance, which pays more attention to the angle rather than the magnitude. If you can use, for example, cosine distance if you think that things can just be scaled up, if like 1 is the same to you as 10 because the ratio between the features is the same, so you don't care about the magnitude, so you can use cosine distance. But what's probably more important is how you turn the distance, or something you have to play with more is how you turn the distance into a similarity. When you turn your distance into a similarity, let's say the bandwidth of your Gaussian is too big, like it looks kind of like the Gaussian that they drew here is quite big, then everything will seem similar to everything else, and it doesn't really tell you the structure of the graph real well. So if you use that kind of too big bandwidth, you'd have all these connections that are cutting across the Swiss roll in kind of a weird way, and you wouldn't emphasize this shape. So the kernel you use to go from the distances to affinities is always going to be really important, and is going to determine a lot of different things. If you made it too small, like if you made the bandwidth so that this is like a little blip, then you would get a disconnected graph where things aren't connected. The points seem like islands. And so then you wouldn't understand the kind of paths through the data or clusters either. So usually you want to set the bandwidth so that you have a variety, that some things are close and some things are far. And this also gets into the idea of perplexity, which we'll talk about tomorrow. But that's the idea of setting the bandwidth. Sometimes people set the bandwidth to the standard deviation of the data. Within the data. There's other ways of picking it. You can also pick it sort of by trial and error. Trying to see if there's any other questions. How about features? What?
I was just going to say, there are a bunch of questions in the Slack that thankfully the TAs are being very aggressive about replying to. So
that's fine. I just wanted to go over a couple just still at the.
No, 100%. No, I think it's good that you are. I'm just letting you know that also there are questions in Slack too if you want to take a look later.
Sure. Somebody asked in Slack, how about features? Do we assume to include all genes in practice when we build our graph? That's actually a really excellent question. No, you don't have to use every gene. That's up to you how you determine the distance. Sometimes we're only interested in looking at certain processes. For example, I know TA Arthi has been looking at some cancer data, and she's specifically interested in metastasis, so she subsets to those genes. You could do that. If you felt like some of your gene measurements weren't good, or you don't want to regard them, you can take them out. And also another thing you can do is you can weight your features. Sometimes in single cell measurements like mass cytometry, people carefully curate the features that they're measuring. And some of the features are really much more important than others. So for example, lineage markers, like is this a CD4 cell versus a CD8 cell? It's a lot more important than, you know, maybe some signaling marker. And they weight it that way so that all the T cells end up together, or all the CD4 positive cells end up together. So there's a wide variety of ways you can use the features, but the end result idea is that if you turn this representation from the tabular matrix to the graph, you'll get something where you can work with it a lot more. Okay, here's an example. Here's the example I was showing you. So this is the distance matrix, and it sort of has these 3 bands. And the reason it has 3 bands is things are somewhat close to points on the other side, other band, as the roll is coming back across. So things on this arm are somewhat close to things on this arm. That's what's creating this side band here. But one advantage of turning this into affinities that are properly tuned, or a nearest neighbor graph, is that you cut out these kind of spurious edges and you're really going through the data. And it turns out manifold learning is all about learning the intrinsic shape of the data and going through the data. The reason is because you're assuming that if your cells are organized this way, then this is a progression among your cells. So your less differentiated cells might be here, more. And these cuts across the manifold are because the intrinsic shape of the data is coiled. And there could be maybe no real cells that could exist here. That's maybe not even a viable state in the intrinsic state space of this cell. So one of the advantages of going to a nearest neighbor graph or an affinity graph with a properly set bandwidth is that you emphasize the connections that are going through the shape of the data rather than spurious connections that happen due to the nonlinearity or coiling of the data, if that makes sense. And you can see that pretty easily on a Swiss roll. And this is one of the reasons that the shape is really emphasized in these. Okay, here's another example, just to give you another example. This is not a Swiss roll, it's a U-shaped manifold that Dan created. So this is the distance matrix. So you see the distance matrix has certain kinds of artifacts, like the blue points are similar to the red points. But within the intrinsic shape of the data, the progression is definitely going from blue to red. So so you see in the distance matrix that both a diagonal and an off diagonal have low distance. However, when you convert it to an affinity matrix, you lose that effect, and it's only the diagonal and some things that are close to the bottom that have high similarity. And the way we got that was because we took these pairwise distances. These are pairwise distances between these points. So self distances point to itself, and then every pairwise distance organized in this order. We're just coloring it by the magnitude of the distance. Now we put it through this Gaussian kernel to get an affinity. When you put it through the Gaussian kernel, it really localizes it, so things are strongly connected to only a few of their neighbors. And now this is a graph, and this is a graph that's emphasizing close connections and therefore, sort of by induction, the shape of the data. And this is kind of the representation that we'll use for a lot of the methods that we're doing in the class. So you might kind of ask why. So why is this kind of graph, this kind of affinity graph or nearest neighbors graph useful? Using a graph as this kind of intermediate representation can make a lot of the analysis tasks that we're interested in a lot easier. So look at this graph, for example. In this graph, you can tell pretty easily that there are 4 different modules. First off, because this graph is laid out nicely, and that's visualization. So graph layout can be a type of visualization. Secondly, you can usually tell this analytically as well, because you can run what's called a min-cut algorithm. You can try to see, how can I bisect this graph using the minimum number of cuts? And you'll get the cuts that are right here, because if you cut here, you're cutting more edges. The minimum number of edges you can cut goes right through where the graphs are naturally split. So actually, you can use this kind of minimum cut to cluster the graph in a way that you couldn't with the data point cloud. And so min cut on a data affinity graph was the first sort of clustering, which is called spectral clustering. Later, people would do another kind of clustering on graphs called k-means, which we'll go over in one of our lectures. Another reason, so you can cluster data more easily from a graph representation, for sure. Another reason is you can understand paths of progression, trajectories, or differentiation more easily. Basically, the idea is you take a walk on a graph. So if you start walking on this graph, you'll get a path through the data. In tomorrow's lecture, we'll talk about diffusion, which is a probabilistic way of taking a walk through the graph. And you're going to compute probabilities of getting from one point to another. And that will emphasize very major pathways in the data. So that's another reason why you want to represent it as this kind of graph, because every time you're at a point, you can tell what other points the point is connected to by an edge, and you can choose to go to the strongest edge, for example, with higher probability, and that'll take you on a walk through the main path in the data. And that way you can understand trajectories in the data. Okay, and then there's much more to come. In the rest of this class, we'll show that the basic idea of turning a data into an affinity graph leads to visualization, clustering, denoising, pseudo time, and many other kinds of analyses, which we won't go over today, but these are all important tests. You want to eliminate noise from the data. You want to understand the structure of your data, whether there's pseudo time, which is a way of analyzing progressions or clusters, and they'll all use graphs, so it's an important concept to come across. So now, so hopefully you understood a little bit about representing data and changing representations, which is a lot of what we're doing in unsupervised learning. And now Dan will talk to you about pre-processing.
Awesome, thank you, Samantha. I also just want to address a bunch of people have been asking, I think, really great questions in Slack about like, how do you pick the bandwidth of a kernel? Why do you use this kernel or another kernel? Like, what's the effect of changing K and stuff like that? And those are all amazing questions, and we are going to talk about them. We're going to address basically a lot of those kinds of questions throughout the workshop, because the answer to those questions is going to change if you're clustering versus visualizing versus doing trajectory analysis. Like, all of the parameters maybe have different sort of selection criteria that are very task specific. And a lot of the notebooks are going to be, have exercises like, try setting this value here and then set it there. What do you notice is the difference? How does that change how you think about it? So it's really great that people are asking those kinds of questions. And I really encourage you to continue thinking about them as you go through the coding exercises. Okay, so the next topic for the workshop is going to be, we're going to talk a little about how to actually pre-process and analyze some single cell data. I think, this is machine learning for single cell analysis, and a lot of the things that we're going to be doing involve single cell RNA sequencing data. And the first half of the notebooks today are going to sort of address some of the things Smitha was talking about in terms of graph construction. And then the second half, we're going to focus on actually loading a single cell data set and then getting into some of the nitty gritty of doing pre-processing. Because as Smitha mentioned, when it comes, when data comes off of the sequencing machine, you get one representation of the data, but there's actually some work that needs to be done to go from the output of the sequencing machine to the input that you have to, say, building a graph. To use a sort of a scaffold for this part of the presentation, we're going to use a tutorial that was published in Molecular Systems Biology about a year and a half ago from Malte Lucan and Fabian Theis, both really amazing scientists working on single cell methodology. This paper, I think, is a really great way to get sort of a first sense of like, how do people sort of think about and talk about different approaches towards actual pre-processing and analysis of data. Understand, actually, I just had a meeting with Malte this morning. He, wants to make sure that everyone knows that these are not hard and fast rules, and that there will be recommendations in this article that change as we start to understand a little bit more. So please don't, take this as a, if you don't do it the way it's described in this paper, you're doing it wrong. These were just sort of what one set of authors thought was the right way to do it back in the summer of 2019. So when data comes off of a single cell sequencing machine, there are, basically four kinds of pre-processing steps. There's sequencing and read mapping. So if people are somewhat familiar with genomics, right, you have a reference genome and a set of reads. You need to align those reads to the genome. If you're using a 10x Chromium single cell sequencing instrument, there's a program called Cell Ranger that does all this for you. There's then a step of quality control and filtering. This is, making sure, I showed you a lot of, if you remember that image of the droplets at the beginning, there was, for maybe 10 droplets getting sort of dispersed into the emulsion, there was one that actually had a cell in it. And so one of the steps we have to do is actually make sure we remove the cells that don't contain healthy cells. They maybe have some ambient RNA floating around in the media or in the buffer. That's actually going to get sequenced, but doesn't contain a cell that we're going to use for our analysis. There's then a step of normalization and data correction that we're going to talk about. Data correction is more going to happen tomorrow. We're going to talk about normalization in the next set of slides. After the sort of basic pre-processing steps, the next thing that people often do is visualize the single cell data. And we'll talk about visualization is going to be a major focus of tomorrow. And then, actually, sorry, data corrections on Wednesday, so 3 days from now. Visualization is tomorrow. And then after you sort of understand the data quality, the next step is to actually do some downstream analysis and maybe things like clustering, trajectory inference, differential expression. Maybe you have multiple conditions. You want to understand the differences between them. There's a lot of other things you might want to do down the road, but we're going to spend today mostly talking about the first set of preprocessing. So as I mentioned, step one, I'm going to sort of walk through this. We have our sort of process here. We're going from some set of biological samples and a sequencer. We get a bunch of reads that we then align to a reference genome, and then create a set of counts matrices. We showed these earlier. It's rows correspond to cells, columns correspond to different kinds of genes, and you get maybe one of these per sample. We're not going to actually talk that much about how you go about doing this. There's a lot of different kinds of sequence alignment algorithms, but most of this time where machine learning actually comes into this process is much later. And so we don't, we sort of assume that you have a set of count matrices already. And then the next step is that you need to do some filtering. So I mentioned before, there might be some empty droplets that you have that you want to remove from your data set. But that's not the only source of sort of noise, or you might think of these as rows of that counts matrix that you want to remove. These are cell barcodes. These are individual beads that correspond to a single droplet that we maybe don't want to consider for downstream analysis. We need to remove them. So for example, you might have dying cells, right? In order to do single cell sequencing, it's great if you're looking at cells from, say, the peripheral blood, where cells are already in single cell suspension. But if you have a tissue, especially imagine a neural tissue where there's a huge amount of extracellular matrix, there's a bunch of support cells, you have these neurons with really long axons that are going to have to get then disassociated into individual cells. This is an incredibly traumatic process of dissociation, and a lot of cells die during that process. And even if they haven't necessarily died during the dissociation, they may be in the process of dying. Apoptosis may have been triggered, and these cells may be expressing either different kinds of Cas spaces, or I think I'm going to talk about this a little bit later, but the mitochondria actually starts to degrade, and there's mitochondrial RNA that might get measured, and we can actually look at the expression of mitochondrial RNA to determine if a cell is actively dying, and say, we don't want to use those cells in our downstream analysis. And then finally, it's possible that you have multiplets. If your tissue dissociation didn't go perfectly, it's possible that you have 1, 2, 3, 5 cells all together in one droplet, and you think that they're a single cell, but actually there's multiple of them. And so you might want to remove those kinds of cells from the data set. So I mentioned sort of one thing already, which was mitochondrial RNA, but I really love if people can maybe go to Pull Everywhere and just add some, things. If you maybe heard about this, worked with some single cell data before, just to, mention what do you think we could look at in the data set to actually do some of this filtering, to actually sort of look at what's happening here. I'm going to wait a couple of seconds for some people to total RNA count. Yeah, I think that's, oh wow, we got a bunch come up really quickly. I think people can actually vote on these. Yep, mitochondrial RNA number of counts is a good one. Ribosomal genes, somewhat. I think it's a little bit hard because cells are just larger, maybe have just more ribosomal genes in them. Senescence markers is definitely one we look at. Library size is a huge one. You can imagine if you have two populations of cells, and one of them is twice the library size of the other, and then you have 3 times, that might be indicative of a doublet or a triplet, for example. Yeah, number of unique reads is another good one. If you have a lot of ambient RNA and say an empty droplet, maybe there's only 10 unique reads. Actually, that's usually part of the preprocessing pipeline that 10x does. They actually try to predict, what are the you may in a data set have 250,000 droplets that are sequenced and only 10,000 cells. And so they want to discriminate those 240,000 empty droplets, and they do that based off of the number of unique reads or UMIs. So these are all really great ideas. It's, sounds like you guys are all really thinking about these sort of questions. It's really great to see. So the first thing that, as you mentioned, that we look at is something that you might call count depth or sequencing depth. So this is a histogram where you sort of the data has been binned. The x-axis represents the number of UMIs, unique molecular identifiers in the data, number of unique molecules that were sequenced, and then the y-axis here is the number of cells within that range. So what we can see is that here there was a threshold created where any cell that had fewer than, say, 1,500 unique molecules per cell are removed from the data set. So those are seen as more of the empty droplets. Those are the unique reads we were talking about. And looking at this data set too, you might want to see, most of the cells are somewhere between 1,000 and 10,000 UMIs per cell, but there's also a distribution of cells centered at 20,000, and then an even smaller population, and maybe a little bit hard to see depending on how big your window is, but there's a smaller population centered at 30,000. So you have to be a little bit careful when you're filtering larger cells. If your sample, for example, is a tumor that has infiltrating lymphocytes, it's very well known that tumor cells are much bigger, have a lot more RNA than T cells. And so your T cells might have really low library size, and then your tumor cells have really large library size. And maybe you want to remove them, maybe you don't. But I think, just keep in mind that if you have some reason to think that there are actually a population of cells with twice the library size of some others, go into the data set, characterize them, always lean on the side of removing data after you've looked at it and see that those cells, oh, it's actually the same set of genes that are expressed just twice as much, or, oh, interesting, there's some markers that I know should not be seen in the same cell, like B cell and T cell markers, but I actually see them together here. Those are cells I should remove. So those, I think, are really good indications that those might be droplets. Someone asked, how do you pick the red line here? And as you're going to hear as an answer in a lot of the things that we're talking about, this is much more of an art than a science. You can think about, looking at distributions and trying to say like, well, it looks like there's a little bit of a dip here between, 1,200 and 1,400, and then it goes back up again. And so you can imagine just seeing where that dip is, then cutting below it. That's similar to what would happen if you use, for example, a Gaussian mixture model, would try to actually just do that implicitly. But honestly, the answer is it gets eyeballed a lot and then verified against other things like the mitochondrial count. So, or, for example, we might look at, the number. So here we're saying, look at also the number of unique genes. So if all of those reads map to one gene, that's not necessarily a good thing. You want sort of you might think of this as sort of the complexity of the data set. I mentioned sort of looking at empty droplets, and this is a plot that I'm just showing. I don't actually like this plot. It's very confusing. Each point here is a cell. This is the rank in terms of the library size of that cell, and the y-axis is the count depth. If you use Cell Ranger, you might have seen a plot like this. And the idea is that the algorithm tries to find a place where this curve starts to plateau, and as you go in increasing barcode, you get a huge as you get increase in rank, the number of UMIs per cell goes down dramatically. And so where it starts to really fall off. And so here you would keep all of the cells above here, say, this might be actually our 1,500 UMI, actually this would not be 1,500, but somewhere around there. We could start to look maybe 11,000, 1,100 or something like that. Finally, we might want to look at a plot where we have, so here this is a scatter plot. Each point is a cell. The x-axis is the count depth, the number of UMIs or reads per cell. And then the y-axis is the number of genes detected in that cell. We see actually that there's some interesting patterns here where there's some cells that as you go up in as you go up in count depth, the number of genes doesn't increase. So you call this really low complexity. You might want to remove these cells from your data set. Whereas most of the data sort of falls along this line, whereas you sequence more genes, your count depth increase, as you sequence more genes, you get more reads, sequence more reads, the number of genes that you're detecting increases, which is what we would expect to see. And then you might also do something like take this plot and look at, say, the number of mitochondrial counts in the data set and look at the fraction of RNA mapping to the mitochondrial genome. as I said before, this is much more of an art than a science. I know that I haven't gone into too much detail here just because we're limited a little bit on the amount of time, but I want you just to know that you can consult that manuscript. There's a bunch of other papers on low-level pre-processing. I encourage you to consult, and we're actually going to play around with some of these thresholds later today in the notebooks. We'll hopefully give you a little bit more of a sense for it. In the last 10 minutes today, I want to start to talk just a little bit about normalization and transformation of the data. So when we talk about normalizing data, we're often thinking about this measure of, we have this big underlying question of similarity. I have a bunch of cells. I only know what genes I'm detecting in them, and I want to know which are similar. I'm going to tell you a story right now of 3 different cells. We've got 2 blue square cells and 1 purple triangle cell. And the first blue cell, let's imagine that we're capturing 10% of the mitochondrial, sorry, not mitochondrial, of the total messenger RNA in the cell. So we have here a cell A, and we're measuring 3 different genes in it, X, Y, and Z. And maybe we detect 10 transcripts of gene X, 20 transcripts of gene Y, and 70 transcripts of gene Z. So what we would really like to do, is understand then, here we know that this is the capture efficiency, but we want to understand what's the ratio of those expression levels within that cell, right? I think that, and I'm going to express to you that this is that ratio is maybe more important than the absolute numbers. And normalization is the process of expressing things as a ratio, instead of just the total counts. So we might imagine another blue cell that we get, and just from some random reason, instead of capturing 10% of the RNA in the cell, we capture 20% of the RNA in the cell. And so, imagine it's exactly the same cell, and the counts we get for each of those genes is totally doubled. So it's just, 10% to 20% go up by 2. So the counts that we get for each of these genes goes up by 2. And then let's imagine that we have another cell, our purple cell, and it expresses an entirely different gene program. Let's say we get maybe 20% capture efficiency on this cell, and this cell maybe has fewer RNAs in total than the blue cells. And what we're getting here now is expression of counts that are somewhat similar to the values in each of these, but looking at the capture efficiency, the ratio between them is very different. For example, gene Y isn't expressed at all in purple cells. So if we just take the sort of raw counts that we're getting out, you see now I've just transposed that matrix before into a format that corresponds to the counts matrix that we get. So this is a very simplified counts matrix. And we take those Euclidean distances that we were talking about before as we're building a graph. We take for each cell, we take the difference, let's say we want to take the distance between cell A and cell B. We look at expression of gene X, subtract it from the difference in gene Y, gene X in cell A minus gene X in cell B, square it, plus the square of differences in Y, plus the square of differences in Z, take the square root. What you get as a distance between those 2 cells is 71.4. Now the units of that, there's no units, it's a distance. You think about maybe UMIs is the distance of 71.4 UMIs or something like that, but just know that the distance is 71. And then if we take the distance between A and C and B and C, we get these following set of pairwise distances. And what I hope is apparent looking at these numbers is that A and B are about as similar as B and C are. And that and this is actually, I think, really bad. You would like to have A and B be very close to each other, and then A and C and B and C be far, because these 2 cells are the same cell. They just have different values because the library sizes of the cells are different. One had 20% capture efficiency, one had 10% capture efficiency. But because we're just looking at the raw UMI counts, the Euclidean distances actually say, for example, that A and C are closer than A and B are, which is not good. And then that B and C are about as similar as A and B are, which is not good. We would like to have A and C and B and C be much greater. And there's actually a relatively simple solution to this. If we just take the library size, the number of UMIs per cell, you can see here, these are that's just the sum of the rows. and then divide those counts by that library size, now what we get is expression of each gene as a proportion of the total library size. You could multiply this by 100 for each cell to get the sort of UMIs per 100 hundreds, reads. But this is a proportion now. And if you look at these proportions, what you find actually is that the distance between A and B is 0. They are the same cell. And then the distance between A and C and B and C are the same and greater than the distances between the two cells that are the same. So I hope that, it's just clear looking at this, that by normalizing by the library size, we can take into account the fact that we have different kind of capture efficiencies from these two cells and actually get the correct distances out. And this is really important because if you had a graph, you might be saying that A and C, or B and C were really similar in that graph, and we don't want that. We want A and B to be very similar. I'm not going to talk about the details, but this is one normalization scheme where you just divide by the total library size. There are much more complex approaches to doing library size normalization. This is a method from a tool called scRNA. It's discussed a little bit in that best practices tutorial. If you're interested in learning more, we're not going to really talk about it today. There's a number of these different strategies. And I would suggest that normalization is not just one thing, but there are a couple of ways to think about it. Finally, I want to mention transformation and scaling. This basically says that, we know there are some biases in the number of transcripts for different kinds of genes. Some genes like actin and tubulin, which are responsible for the underlying structure of the cell, are expressed at higher levels than maybe a transcription factor, which you maybe only want a little bit of at a specific point in time, but you always want a lot of actin in the cell to make sure that the cell sort of maintains its internal structure. And so you might not want, Euclidean distance takes into account the absolute difference between two numbers. And so if you have one gene that is very highly expressed and another that's really lowly expressed, differences in the highly expressed gene might be greater because, say, have more wiggle room in terms of the total number of UMIs that you can see than a transcription factor, right? It's easy to imagine that there might not be a huge difference between 1,000 and 1,100 UMIs of actin, but there would be a huge difference if you're looking at, say, Wnt, and you have 1 or 100, right? 100-fold increase in expression in another cell. So there's a process called transformation where you, for example, take the square root of all of the numbers to just get them on a little bit more of an even playing field. So distances between them are a little bit easier to understand. So one of the ways you could do this transformation is using square root, but sort of the last question of the day I'd like to add, well, actually there's one more quick question at the end about just general questions, but can some people suggest, spend a minute here, what are other kinds of transformations that you could do? Yeah, so great. I mean, the two big ones are log and z-score. These are absolutely right. And there's a bunch of kinds of log 2, log 10, natural logarithm. SC transform is actually also a way to do this, which is based off of, it's sort of a variation on z-score with some sort of local regression for each of the variables. I'm glad to hear that everyone is familiar with a number of different kinds of ways of doing this. It's important to note that all of these have caveats. So like one of the things that log has a problem with is if you have a 0, you cannot take the logarithm of 0. And so what people often do is add what's called a pseudo count. They just add 1 to every single value because log of 1 is 0, and then you can, some logs of 1 are 0, but you can then account for zeros in your data set. This is a bias, right? You're adding 1 to everything. So that means that you're adding 1 to a gene that is expressed at a value of 2, and you're adding 1 to a gene that's expressed at a value of 1,000 UMIs. And that will bias the data, and it's going to bias the data more for genes that are lowly expressed. That's one of the reasons why we like to use square root, because square root is not does not have this property. You can take square root of 0, but square root is sublinear and has other sort of complications that arise from using that particular transformation. Again, we're not trying to say that this is like necessarily the right way to do it, but just the way that we sort of approach this in the lab. And there's a lot of different ways to think about it. And I hope that the message that you're getting from me saying stuff like that is that there's a lot of ways to do this that are equally valid and that have sort of different, approaches. The final thing that maybe counts as pre-processing is called highly variable gene selection. This could also be considered a part of dimensionality reduction. The basic idea is that here each point is a gene, not a cell. The x-axis is the average expression of that gene on a log scale. And then the y-axis is the standardized variance. And the idea is that there are some genes that are more variable than you might expect based off of their mean than other cells. We're going to talk about this a little bit in some of the notebooks, but you might have a process like calculating this mean expression and variance. You fit a lowest curve to this plot, and then take the maybe top 2,000 or 5,000 highly variable genes. So this would remove things like actin or that is sort of uniformly expressed across cells, and keep more interesting transcription factors that probably have higher variance across the data set. The rest of the workshop is going to focus on teaching you the things that come after this pre-processing. Today, we're going to focus mostly on the sort of topics that I mentioned now, but I really encourage you to think like, how, if I change some of these things in pre-processing, how is that going to change the final results of these algorithms that I'm looking at? People have been actually really good about asking questions on Slack and in the chat. And so I think actually instead of taking too much time right now answering specific questions, I would really encourage people to post questions in the chat. We said we were going to end the lecture at 1:30 and, or sorry, not in the chat, on Slack. And there's a MISC help Slack, there's a math help Slack. If we answer and ask questions there, we'll make sure that they get sort of preserved, and you can maybe get some more rigorous responses from multiple TAs and submit to questions that you may have. So this concludes the first lecture of the workshop. You made it through it. I know that was a long time to be sitting down. We're going to jump right into the office hours section. The office hours, I want to emphasize, are optional. We're not really, this is, I think, an opportunity for you to get the help, the sort of benefit of this workshop. So although, you don't necessarily have to be there, we have a, cast of 17 TAs who are going to be here to answer every question that you have for the next few hours. And then afterwards, it's going to be a little bit harder. So I really encourage you to stick around. This is, I think, the real value add of the workshop itself. So there are 2 tracks. Everyone should have know which group that they're in and what the level of their group is. And so what I'm going to do is open up the breakout rooms in a minute. And there's going to be basically 2 things that are going to happen. So for everyone who's in the advanced group, you were put in the advanced group because it sounds like you're familiar with Colab, you have a little bit of Python experience. I really encourage you to go into your groups, introduce yourselves to each other, your name, where you're from, why you came to the workshop, what you're excited about to work on, and then start with the exercises on the workshop website. For people who are in the beginner groups, before you go into your breakout rooms, we're actually going to have a little tutorial right now from Scott, who's going to open up one of those Colab notebooks and just sort of introduce you to like, how do you run code there? And how does Colab sort of work? And make sure that you understand exactly what's expected of you in those sessions. And so, I would encourage advanced groups to go into your breakout rooms. And then I would encourage the people in the beginner groups to stay here. And we're going to send you into your breakout rooms in a little bit. So, we'll maybe start Scott's tutorial in, let's just say, like 3 minutes or so people can go use the restroom if they need to. And with that, we're going to open up the rooms. You may be, so some people are assigned to rooms ahead of time. We've had issues with Zoom breakout rooms in the past. Just feel free to come back into the main room if you have a problem with that. And if you're in the wrong room, or like the room you're supposed to be assigned to, you're not in. And then, but I think if you have updated your Zoom client to the most recent version, you should actually be able to switch around rooms yourself, or to go, to assign yourself to different rooms. I see right now that there are like 73 people who are unassigned, which means that you logged into Zoom today using a different account on Zoom than the email address that you use to register for the workshop. So I really strongly encourage you to select the rooms for yourself. And if you have if people are struggling with that right now, please unmute yourself and let me know, because Zoom changes every week, and things that tested a month ago may not work right now. So some of the people here, can you actually, everyone here is in a beginner group, is that right? Is there anyone here who's in an advanced group and is having a hard time getting into their group?
How do we know what group we're in? What type?
Yeah, so there's a spreadsheet. There's a group assignment spreadsheet that should say, oh, Scott, does it not say what the level is? I'm realizing actually it might not say that. I think
it does say. I just accidentally clicked on close all rooms, unfortunately, so we're gonna have to reopen those. Sorry about that.
Oh no. Sorry about that, everyone. Can I stop that?
It doesn't look like it.
Oh no.
That was silly.
Well, it's fine.
Oops.
We have a number in the end of group name. Yeah. So actually it doesn't say on that Google sheet what the level is. Oh, this is from last year.
Yeah, it should say.
So if you go into the announcements channel, I pinned a post to the Google sheet. If you go to 2021 workshop announcements, I'm gonna post that group assignments. Yes, and it does say. Okay, so can you see, I think it was Cody had a question. Can you, do you see that?
Yep.
Okay, great.
Thanks.
So hopefully you should be able to look there. Okay, let's see. So sorry, there's an issue. Oh, Scott reopened the breakout rooms. Perfect. Great. Okay, Scott, do you want to start by sharing your screen and giving a start to the tutorial?
Yep, absolutely. Great. Everybody can see my screen?
Yes.
Great. Okay. So I'm going to go to krishnaswamilab.org/workshop. You can also get to this, I believe, in the Slack. It's in the announcements channel. And
I'm
you'll
sorry,
see,
how do we get to the breakout rooms if we are using a different email on Zoom?
So if you should be able to see, can you not see breakout rooms at the bottom of the Zoom client?
No, I can't see that. I'll try to play with the settings a little bit.
Okay.
otherwise, Dan, could you just manually assign people who ask in the chat? Is that the easiest way?
Yes.
So if you can't see the breakout rooms at the bottom of your client, just post which group you're supposed to be in the chat, and one of the hosts will move you.
Okay.
Okay. For everybody who's in the beginner groups, I'm going to run through just really briefly how to use these notebooks. So you can go to krishnaswamilab.org/workshop to follow along with me. So you'll see below the course description, we've got this syllabus that lists all of the lectures, as well as all of the notebooks that you'll be working with and the answer keys for those notebooks. So what I'm going to do, I'm going to open up this one, 1.0 Learning Graphs from Data, and it will open a Google Colab notebook. If you've used Jupyter notebooks before, this is quite similar. So the basics, very basics here are, you can hit Shift Return. Let's go down to a code cell here. Hit Shift Return to run code. And the first time you do this, it'll give you a little warning, just say run anyway. And all you'll need to do in these notebooks, there'll be small sections where we've asked you to fill in the blanks. And if you read through both the text and the code, you should understand what's going on and be able to fill in those blanks. A couple of technical notes. There's this installation block here at the top of every notebook. It looks like we no longer have to do this because Colab has updated their software, but sometimes it may pop up a warning that says you need to restart the notebook in order for these installs to take place. If after running this, you have any messages that are like scprep not found or any other packages not found, you can go up here and go to, where is this? Runtime, restart runtime. And if you do that, the installation stays, but it refreshes the notebook in a way that then hopefully gets around that module not found error, just in case you find that. So strongly encourage you to read through all the text here. Dan and I and a few others have spent a lot of time building this so that it should be fairly easy to follow. And what you're going to do is we're going to run each of these exercises where there are a couple of bits that you need to fill in. So where's an example? So there's the first exercise down here, exercise graph parameter tuning. And so what we're asking you to do is in your groups, there are a couple of different choices here. So for example, if I scroll back up, we have these data sets in this example where we have circles, moons, blobs, and 3 different types of blobs and a uniform data set. And each of these has slightly different structure, slightly different requirements for analyzing it. And in your groups, you'll choose different data sets, analyze them separately, and then come back together and discuss. So in this exercise block, it'll ask you to copy and paste some code from above and modify some pieces to fit the data set that you're working with. Now, there are a few other things that you might need to know. So for example, if you want to know how to use a particular function, there are a few different ways you can do this. First of all, you can type help and write the name of the function inside help. And that will show you the documentation for that function. So here, for example, I found this make blobs function, which has a few different parameters. You can tell it how many samples you want, how many features, the standard deviation, and a few other things which we don't really need to play with. And it'll tell you what each of those parameters do. If you want more help than that, there are a few different things we can do. In some of the, let's see, if in some of the code, we actually point you to some documentation. So there are 3 things that I'd encourage you to do if you're having a hard time. The first one is in a lot of these sections, and for example here, you'll see our lab has developed a toolkit for analyzing graphs called Graph Tools. And if you want to know anything about this, you can click on these links and they'll take you either to the code base or in a later section, where is this? I mean, later section here, we say consult the following link, more information about dataset parameters. And this link will take you to the scikit-learn documentation for this particular function. And so frequently we'll actually give you the links to the documentation. And you can see, for example, this make blobs, you can follow that link and it'll give you something similar to what showed up when I asked for help make blobs. The final thing you can do, if there's a if there's a function that you don't know about and you can't find the documentation for it elsewhere, you can just take the name of this function. So for example, here I have scprep.plot.scatter2d, and we can just Google it. And the first thing you'll see that comes up is this scatter plot gallery, which is not quite what I'm looking for, but you can see there's a lot of different uses of scprep.plot.scatter2d. What I'm really looking for is the documentation here. And that's also the source code reference. Plotting, and here's the scatter2d. So you can feel free to Google for those documentation. If you can't find them, the third thing you can do is ask on Slack. You can either ask directly the TA assigned to your group, and I believe you should be able to see who that is. But there is a TA in every group on Slack in your group Slack channel, or you can ask in one of the help channels if either they're not available or if your question is maybe commonly of interest, you think, to everybody else, or if your TA is unable to help, we can always jump in. So those are the really big ones. The last thing is on the workshop page. If we go back to workshop main page, you'll see there is an answer key. So after you've given this a real shot, encourage you to look at the answer key and see how we did it. If there's anything you're stuck on, please do come to us rather than just checking the answers